Hey guys, today we're doing an A-level biology P5 video, and the paper we will be doing this time is October-November 2010 P51. And again, I will emphasize that you only have an hour and 15 minutes for this paper, and therefore utilization of your time is a key skill you need to develop. Right. So, the most important skill when doing P5, read the question with the utmost focus and dedication. Do not miss out even a single implication. It may have a huge impact on the questions and the difference between an A and even a C. So a student noticed that the leaves on a plant growing close to a wall had two sorts of leaves. So the location may be significant, so don't let it be considered as unimportant by your brain, because your brain processes stuff and leaves out the things it considers to be insignificant or irrelevant, and when you have to use up all the knowledge you have gained while reading the question in answering stuff, you feel at a loss because you don't get what's happening. So the leaves next to the wall were in the shade and looked different from the leaves on the side away from the wall that were exposed to the sun. Right? So the difference between shade and sun leaves, I believe this question has been asked rather frequently in both O4 A2P and in the AS practical part of it. So the length of the internodes on the stem also looked very different. So in addition to the appearance of the leaves being different, the distance between adjacent leaves, which was the distance between the internodes, was different for the two leaf types, or the two parts of the leaf that were two parts of the plant that were exposed to different conditions. And the student decided to investigate the difference by measuring some features of 30 leaves and internodes from each side of the plant. So 30 leaves were considered for each side of the plant. And how were the differences investigated? By measurement of certain features and the distance between the internodes. So the internodal distance wasn't the only thing considered for the 30 leaves of each side, but other features, certain other features were also measured and therefore considered when making comparisons. And this is basically the general leaf shape. This is what both the leaves generally look like. Obviously there are differences, they've stated themselves with the general appearance. And this is what an internode is, and this is the internodal distance. This is an internode and this is the internodal distance. And table 1.1 shows the students' results. So compare for shaded and exposed. Mean internodal length is given in millimeters, 23 and 15. So you observe that for the shaded leaves, the mean internodal distance is greater, meaning the distance between adjacent leaves is greater, right, on average. And what is plus minus 4 and plus minus 3 is likely the standard deviation. So 23 minus 4 is 19 and 15 plus 3 is 18. As you can observe, when considering the standard deviations, there is no overlap. And this means the difference in the mean internodal length is likely to be significant. And then there is a mean surface area of the leaves, and this is given in millimeters square. So the mean surface area, the average surface area. In the case of the shaded leaves, it is 2750 compared to 1800. Once more, there is a definite difference and there is no overlap with or without considering the standard deviations. And the standard deviations aren't that huge to begin with, considering that 12 isn't very huge compared to 2750. So mean mass, 50 compared to 60. Now this is interesting. You'd note that mean internodal length is larger, mean surface area is larger for the shaded compared to the exposed, but when we come on to the mean mass, the mean mass appears to be smaller for the shaded leaves compared to the exposed. And when we consider the standard deviations of 58 and 60, minus 10 would be 50, right? So there is definitely a lot of overlap when it comes to the mean mass, because we can have, when we consider the whole standard deviation, the range is from 42 to 58. You'd better write that down, 42 to 58, because it is plus minus 8, isn't it? Plus minus 8 about the mean position. And then there is 60 minus 10, 50 to 70. So between this range, because it's 42 to 58, 40 to 58, I'm sorry, 50 to 58. So there is an overlap of 8 different possibilities. So this one, the mean mass isn't all that reliable, and compared to this and compared to this, the standard deviation compared to the mean value is more significant as well. And mean leaf surface area to mass ratio. So this is ratio. We have ratio of 55 and we have a ratio of 30. And how do you get this? You basically divide 2750 by 50 or something. Yep. And same here, you divide 1800 divided by 60, and you should know ratios don't have any units. And there is deviation here as well. The deviation is more significant here compared to over here. And let's talk about overlap. So 55 minus 19 is basically 46, I believe. Yep, 46 to 46 to 64. This is quite the range, isn't it? And here we have 30 minus 6, 24 to 36. So there isn't any overlap, thankfully. And then we come on to over here, rate of water loss. There seems to be a difference over here. Here the difference isn't all that significant and there is definite overlap. And the standard deviation values are somewhat huge and therefore significant. So this would be 50 minus 11 would be 50 minus 10 would be 40. So 39 to 61. And 65 minus 12 would be 3 53, 53. Now 65 minus 12, 65 plus 12, 77. So there is definite overlap, at least from the 53 value to the 64 value. So it's not all that significant, but the values do have some significance. So question number 1, we have to state the independent variable being investigated, and that is a thing we can actively change in order to observe, or the change, or the thing, or the factor that is different. And what is that? The difference is the independent variable is whether or not the leaves are grown in shaded or exposed conditions. So the type of leaf you may write. And outline the procedures the student could use to obtain these results. So what kind of procedure the student could potentially use to get these results that have been recorded here. So in these procedural questions, there are a lot of things you need to take care of. You might ask why, and because you're actually giving the procedure from start to finish, so to speak, as if you're dictating the procedure to a complete rookie. So you have to be really detailed. You have to tell everything, how you would initialize the procedure, what sort of instrumentation would you use, what would you use the instrumentation for, what kind of data would you derive from the experimental procedure you have just detailed, what sort of precautions ought to be taken, what sort of things ought to be standardized, what sort of things ought to be made, and the precision and the accuracy of the measurements you have used, right? And stuff you need to be really careful with in order to get accurate results. So what is the procedure to be followed? So we have, as I've already mentioned, that we have taken 30 leaves to be considered from one side and 30 leaves to be considered from the other side. So one thing that is really important is that when you're writing the procedure, you have to be really careful that you explicitly state this point so that they know that you know what sort of scenario you're considering, and the information that is already given ought not be changed in any manner. It should be further amplified or further detailed. So mean internodal length. So we're saying we have to consider 30 different leaves, right? Decided to investigate the differences by measuring some features of 30 leaves and internodes from each side of the plant. So what is it that you do in order to proceed? Number 1, your selection has to be totally random so that you are certain that you are unbiased, and whatever sample you have taken is unbiased and you have not been influenced by something. That is the most significant thing you need to take care of, because you want reliability and you want accuracy and you want precision. So you choose 30 leaves completely at random from each side of the plant, right? So let's move with the easier parts first. So for the mean mass of the leaves, you measure the mass of each leaf, and you must be really careful to measure the instrument used. You ought to use an electronic balance, and your mass ought to be accurate to 1 gram at the very least, right? And I would suggest that the leaf ought to be weighed at the same time after it has been removed, because there may be some change in mass after removal. For example, some of the starch may be converted to glucose or whatever in order to be respired, because the plant will want to remain alive for as long as possible until eventually the starch deposits would have been depleted. So you weigh the mass of the leaves as soon as the leaves are removed from the plant. Easy enough to imagine. And what is something that could affect the mass of the leaves? You must realize that the mass is probably not all that big to begin with. So you have to use an accurate electronic balance, right? And you have to take the mass after the value is stabilized, because when you will put it for some time, there would be a lot of fluctuation, right? And then you'd have to take the masses of 30 of them, and calculation of the mean isn't something you're required to explain in this part of the question anyways. And then they're onto the mean surface area of the leaf. So what is the surface area? So basically all of this part as well as the part on the back, if you add up the area of all of this and the area at the back, that would be the mean total. That would be the total surface area of one particular leaf. So the difficulty is, how on earth would you measure this? Isn't some sort of standard shape, and the shape of the leaves would probably be different. One method is to use a graph paper. You put the leaf there and you draw its outline, and then you measure all of the boxes that the leaf is occupied. And because one box would be equivalent to one certain area, because this might be 1 centimeter, this is 1 centimeter, and therefore this one block is equivalent to 1 centimeter square area, actually millimeter, because that is the measurement, 1 millimeter, 1 millimeter. So that is how you go on about measuring the surface area. You have to be really careful though. And then you just basically divide them. That is in the procedure. So I guess that would work as well. The procedure, obtain. Yep, I think so. It would be good if you give how the mean is taken as well as the standard deviation, or at least make sure that it is clear that you know what a standard deviation is anyways. And for the mean internodal length, like I said, randomly take distance 3 internodal distances and measure them. What would you need for that? And probably because these distances aren't that huge to begin with, you may use a vernier caliper, which has a lot of accuracy of 0.1 millimeter, 0.01 millimeter, I think. That's why you get a good value over here. Again, this is done, and the rate of water loss, milligram per millimeter square per hour. Now this is interesting. So this is milligram of water. This is the mass of water. This is a bit of a confusing thing, because one method of water loss would be the use of a pottometer, which has to do with measuring the transpiration rates of cut leaves, and for that you'd actually have to take the whole branches with the cut end, right? And you'd want just one leaf there. But then you can't really do this after having done all of that, because a lot of water would likely have been lost, and you probably wouldn't have taken a huge complete one branch. So that leads us to the question, perhaps we are wrong. And because the values you would have gotten for this would have been in centimeter cube, would have been in terms of volume lost and not milligram. What this suggests is that every hour this much mass of water is lost for every millimeter square, and that is confusing. So what is mass? Mass is basically the amount of substance, the amount of water, and that is what you measure in milligram or gram. And do we see any of these units around here somewhere? We do. Mean surface area, and we have mean mass of the leaves, and then there is per hour. So what we can do is yeah, there would have been mean mass of the leaves immediately after the freshly cut leaves were taken, and then you may leave the leaves as they were for an hour or two to speak in conditions that are constant in terms of temperature, in terms of the humidity, and in terms of the wind speed, because you want to standardize those conditions, right? So you would get any value of mass because the water would have transpired in this period because of the stomata still being open. So then you would take the difference between the initial and the final masses for each of the 30 leaves from each of the side of the plant. But you should keep in mind that while by taking the difference in the masses, you may have accounted for different initial masses for each of the 60 leaves. The fact is the surface area is likely to be different from each of them, and surface area is definitely something that will impact the rate of transpiration. And so you divide each of the difference in masses for each leaf by the initial surface area, which would still be the same, and you would get the rate of water loss. And because the time is the same, if it is for just for 1 hour, you don't need to divide by anything, but if it is for 2 hours, say, you'd have to divide all of this by 2 as well. And that is how you would get these values. And then standard deviation part is easy. Hopefully I made stuff clear for you. So did I mention the sort of variables that need to be standardized? I did, in terms of the transpiration one. Any sort of precaution? You might want to wear gloves in case you're allergic to some sort of pigment on the leaves that might get out when you cut. So this is basically a low risk investigation. And you may also be careful when you're using some sort of a knife to cut the internodes or to cut the leaves. You may accidentally cut yourself. So again, wear protective gloves and be really careful when cutting the stuff. And have I mentioned all sort of the instrumentation? The specificity of the instrumentation has similarly been mentioned. Yep, and is there any sort of need for replication? Well, you'd have realized that 30 leaves, those are basically just replicates in and of themselves. So I would say there isn't much of any need for any sort of replication. So normally while we do write about replication has itself been stated as part of the question statement, so no need. You wouldn't get any mark for saying that you need 30 more, or like repeat the same thing. Don't. A possibility is that you take the water loss over a longer period, 10 hours, so that you get more accurate values. So the student carried out Ttest for leaf surface area to leaf mass ratio and for internodal length. So what were the Ttest carried out for? So on these two and on these two and in these two. And what does the t-test do? It basically compares the mean of two sets of, so one set and the second set, and this is one set and this is second set, because this is the two sets I'm talking about. Two sets of continuous data, the mean of two sets of continuous data, this one and this one, and it tries to figure out if the difference in the mean is due to some significant difference between the conditions, or is it entirely due to chance, entirely due to natural variation. That is the purpose of a t-test. And the leaf surface area to leaf mass ratio, give the value t is equal to 12.6, and the formula for the t-test is given, and these formulas are often given, so you don't really need to memorize them, but if you do have them memorized, it's often easier. So you need to complete the calculation to find the value of t for the internodal length, and we need to show workings. Right? So over here, so it's x bar 1 minus x bar 2, and x bar 1 is basically the mean of the first set, and because we're talking about the internodal length, here it is, so it would be 23 minus 15, because this is x bar 1 and this is x bar 2 for internodal length. So 23 minus 15, and this one has already been mentioned, and this is for this one first, 4 divided by 30, because 4 is the standard deviation and 30 is the sample size, and to that we will add 3 square divided by 30, because 3 is the standard deviation in the second one, and when we calculate, add up these and take the under root, we get 0.9, and when we minus 15 from 23 we get 8, so 8 divided by 0.9. Take your calculator, how much big of a value you would get? 8.88889. So I think so it'd be good if you got your answer to two significant figures, 8.9, but you can write 8.8889 as the answer beforehand and give 8.9 as the actual answer, but if you mention this, it's a good idea. And this table 1.2, what is it is showing? It is showing the critical value at p is less than 0.05 for the t-test. So so if p is less than 0.05, what does that mean? It means that it is significant. If p is less than 0.05, it means that the probability of the results being due to chance are getting less and less, and therefore more and more significant. So how much degree of freedom do we have? What is degree of freedom? How do you calculate degree of freedom? Right? How do you, because we're dealing with a t-test and there are actually 60 different leaves that you've taken. So 60 minus 2 would be likely be 58. And that is basically just how degrees of freedom work. In the case of chi-square test, it is n minus 1, n being the sample size, whereas in the case of Ttest, you actually have to increase the value. And there's actually a bit about it written in the A-level biology book by Mary Jones, Richard Fosbury, Jennifer Gregory, and Dennis Taylor. And the degree of freedom is n1 minus 1, which will be 30 minus 1, 29, plus n2 minus 1, which is again 29. And 29 into 2 is basically 58. So the critical value will be somewhere in between 30 and 40. So 40 and 60. So it would be 2.012 something. Right? Right? You're getting it right? So state how the number of degrees of freedom was calculated. I just did. And state and explain the meaning of these results. So the critical value is 0.05 for p is equal to 0.05. And something to remember is that when we talk about critical values, when the probability is decreasing, the value of the t-value, the value of the t-value is also increasing. Okay? And that is basically what all of this is. What does this mean? Because we have a critical value of about 2.02 to 2.0, somewhere in between. And whereas the value that we have gotten is 8.88, which is much huger than the critical value. And this means that whatever value we have gotten is most certainly significant, because it is much greater than the critical value. You need to have at least a value between 2.02 and 2.00 for it to be significant. And because the value which we have gotten is much greater than this, then it is most certainly significant. Now we have, in a further investigation, the student cut sections of the leaves from the shaded side and a section of the leaves from the exposed side of the plant. So in a further investigation, the student cut sections of the leaves from the shaded side and from the exposed side of the plant. The following procedures were carried out. Transverse sections were made of each leaf, and high power drawings were made from these sections. And in high power diagrams that you are likely familiar with from AS, you have to include a lot of the finer detail. You actually have to draw the structure of the cells in and of themselves. And so the relative thickness of both the leaf and the cuticle were made using an eyepiece reticule, and the difference in the distribution of the chloroplast was observed. Right? And these are the drawings. And explain how the actual thickness of the leaf could be made. Well, for that we'd have to calibrate the eyepiece reticule using a stage micrometer. We would have to superimpose them at a particular thickness, and relative to that, calibration would be a definite possibility. So with reference to the student's results, state what conclusion can be drawn about the differences in adaptations shown by the shaded leaves and the exposed leaves. And this is a bit of a complicated question. So with reference to the student's results, you have to refer, and what you have to refer about, what references do you need, to what sort of references do you need in order to support what you're writing, and what you're writing is the conclusion can be drawn about the differences in the adaptations. So what can you conclude? You can conclude that the cuticle is visibly more thicker for the leaf from the exposed side of the plant. And what could be the reason for it? We'd conclude that perhaps the difference in exposure means a difference in the amount of water lost, and a thicker cuticle is what would be preventing said water loss. And we can also conclude the thickness. The exposed side of the plant is much thicker than the leaf from the shaded side. That is what we can conclude. For what reason, I don't know. Another thing, the chloroplast seem to be more widespread in the exposed side, whereas in the shaded they seem to be more concentrated at the center of the leaf. And then there is something about the shape as well. These are more longitudinal. They are more long, so to speak, because the light is probably from all of the sides, and we want maximum light, we want maximum surface area to utilize all of that light. And perhaps in the shaded region there isn't all of that light to begin with, and so it's more likely to be concentrated at the center. And then we talk about intracellular spaces. There seems to be less intracellular spaces in the exposed side, maybe because we don't really need all of that, or for the sake of all that water loss, maybe. What can you conclude? So the cuticle is something you're definitely onto. There's a definite difference in the thickness here and over there. And there's something of a diameter difference here and there as well, but not perhaps all that significant to begin with. And then when you consider the mesophyll cells, the palisade, these ones are definitely much more longer and much more cuboidal. These are a bit more squishy. And their length is definitely less. And then there's the whole chloroplast distribution. It seems to be more widespread and maybe more towards the downward. More of them seems to be at this position. Here they're more likely to be either completely widespread or more centrally located. Maybe something about the spongy mesophyll, there are less of them here, and the intercellular spaces is greater, has more volume, less volume. And again, it does seem apparent that the chloroplasts are more towards the edges of the cell over in the exposed side, and more centrally located over there. And perhaps this is due because there's a lot of light from all directions, and so they position themselves in the way that is most beneficial. There isn't any such need over there because there is so much less light to begin with. So definitely write about the cuticle and the chloroplast distribution, because the reasoning seems set enough, and we need at least a third point to get the full marks. What else could they be? And then the central and non-central distribution of the chloroplast, and the differences in the width of the cuticle. And we may also say that there is less intracellular space because we want to reduce transpiration, because there's a lot of transpiration over here. There isn't much transpiration over there, so even if we have a lot of intercellular spaces, there isn't much of water loss, and the plant isn't gravely dehydrated. So we have to state the independent variable being investigated. Light, intensity, या फिर exposure. Do not allow light unqualified or position in the shade or the sun. We messed up on this one. 8 of if we say lack of exposure or exposure, that is definitely a difference whether or not it is exposed to the sun or not. So what is the independent variable? A reference to a systematic way of obtaining the leaves, and ignore any reference to planting seeds or potted plants. We don't want to go that way back. So example, third leaf from the apex, different heights, all from the same height, or all that have this equivalent exposure, or all that are from the same height, or we basically choose them from different heights. Or example, dependent variable, reference to a method of measuring the surface area. Example, draw around each leaf on grid or use transparent grid over the leaf, or measure the diameter of the leaf. Reference to how the surface area is calculated. You count the squares. Reference to idea of both side needed to get total surface area definite. Reference to a method of measuring mass digital balance. Finding dry mass. Example, sample leaves dried in oven until the mass is constant. Reference to a method of measuring internode length, either on the plant or a cut section from a plant, by holding against a ruler or using string or cotton to mark distance, measure with ruler. Reference to a method of measuring water loss. Use a potometer, weigh leaf. This is what we went with. Place the leaf inside a plastic bag to collect the water that is transpired. Refer method of using transpiration apparatus. Measure distance moved by the water, or weigh at hourly intervals, or weigh bag or leaf after a stated time. Low risk investigations, heat and suitable precaution, leaf allergy. Mean values of the whole sample, working out the standard deviation. Surface area, mass ratio. Okay, ignore the formula. So we did fairly well. Let's move on to the actual marking. So this is something we messed on, because the actual variable is the exposure, and we are varying it by putting it either in the shade or the sunlight, and these are some distinct things you need to get an idea for. What you observe is not necessarily the independent variable. So, a reference to a method of measuring the surface area. Example, draw each leaf on grid or use transparent grid over the leaf. How surface area is calculated. Count squares. Both sides needed to get total surface area to a method of measuring mass. Electronic balance and digital balance are basically the same. Method of measuring internode length either on the plant or a cut section. I believe the vernier caliper one is just as right. Measuring water loss. So use a pottometer, I did mention it, or weigh the leaf. That is what I went with. Place leaf inside a plastic bag. You'd collect the amount of water transpired. Weigh at hourly intervals after a stated time. Reference to keeping a constant environment when measuring mass. 1, 2, 3, something I missed out on, 4, 5, 6, 7. So 6 marks guaranteed from this portion. Low risk investigation, 1 mark. Leaf allergy is what we went with, and 1 mark. Actually we did all of these. SA definitely mentioned. Mean values of the whole sample. Yep, so you'd likely get full mark for this. So this whole systematic way one is something you should probably have considered, because in this scenario the different positioning might actually be something that would have an impact, because we are investigating the whole light intensity thing. So even for the different light intensities for the same sample, you want to have similar light intensities, so they should likely be at the same height. And then the whole dry mass thing should have gone with that. Yep, 8.9, 58. Reference, both calculated values are greater than the critical value, meaning both results are significant, not due to chance caused by another factor, light exposure. Counting the number of eyepiece critical units. Finding the value of an eyepiece unit with a stage micrometer. Yep. Marks are for conclusions about the adaptations shown and not for restating what has already been concluded. So 3 off, either this or not. Thinner cuticle increases light penetration. Thinner leaf, shorter palisade cell increase light penetration. Spongy mesophyll have more chloroplasts to increase light absorption. Cells are less densely packed. I didn't write any of this, but this is right. Thicker cuticle limits water loss, and they contain more chloroplast to absorb maximum light. Smaller air spaces reduce water loss, and smaller surface area reduces water loss. Is there a smaller surface area thing going on here? Yep. So 2 done. Unfortunately you would lose 1 mark for this, so for this you would have gotten 20 out of 18, which is pretty good, I think. What did we miss? Larger, longer palisade cells, they contain more chloroplast to absorb maximum light. Fewer chloroplasts as little light penetrates this far. Yep, because most of them would have already been absorbed at this point. Smaller surface area reduces water loss. And that's it. Now let me take a break and then we'll move on to question number 2, which will be more statistically. Yep. And now on to question number 2. Figure 2.1 shows a freshwater crustacean, and the animal has a 2-chambered heart that is visible through the exoskeleton. An investigation into the effect of temperature on heart rate was carried out using this organism to test the hypothesis that heart rate doubles for every 10 degrees Celsius increase. So 5 crustaceans, each measuring 5 millimeter in length, were placed in water of different temperatures and left for 5 minutes for 20 seconds using a tally counter and stopwatch. Table 2.1 shows result of the investigation. So we have 5 crustacean specimens, and they're apparently all of the same length, that is 5 millimeter, and they're placed in different temperature conditions by being placed in water that is at a different temperature, and temperature is 5, 10, 15, 20, 25, 30 and 35. So 7 different temperatures, 5 degrees Celsius intervals, and identify the variables that have been controlled during this investigation. Number 1, the length of the specimen and the time interval for which the heartbeat was counted, and one other variable that could be controlled. So what is something that may have affected the heart rate of the crustacean? Length and time has obviously been controlled. Something else that could potentially affect it, and equilibration has already been done. Maybe the mass of the crustaceans, same length, does not denote the same mass. One feature which may cause the results to be inaccurate, and the sample size, we just have 1 crustacean for each temperature. Oh, because we only have 5 crustaceans, so the sample size isn't all that big. Used 5 specimens because there is a chance of there being natural variation among a species. Different members of the same species might behave differently with the same stimulus. So let's just go over this once more. So we have this freshwater crustacean. We have a 2-chambered heart, and it is visible through the exoskeleton. An investigation into the effect of temperature on the heart rate was carried out using the organism to test the hypothesis, and the hypothesis is that heart rate doubles for every 10 degrees Celsius rise in temperature. 5 crustaceans, each 5 millimeter long, were placed in water of different temperatures and left for 5 minutes. Heartbeat was counted for 20 seconds using a tally counter and stopwatch. 2 variables that have been controlled: time interval in which heartbeat was counted and the length of the specimen. One other variable that could be controlled: mass is definitely a possibility. One feature which may cause the results to be inaccurate, basically we just counted for 20 seconds, so there is a lot of inaccuracy chances. If we had counted for more than 20 seconds, it would have likely been a better idea. And then we have the last part of the question. Then what did the student do? He or she calculated the percentage change in heart rate for each specimen. There was a 50% change, there was a 40% change, 52% change when from this to this temperature. Why the student converted the raw data to percentage change? Because what we are comparing is basically the change or the change in the heart rate for every 5 degrees Celsius rise, and because there's likely to be differences in the initial or the normal heart rate. That is why we are using percentage change, because we want to make comparison easier, more feasible, to see how for each of them the same rise in temperature, how much change is it resulting in. So this accounts for the differences in initial temperature. How the temperature change between 25 and 30 was calculated. So basically 165 minus 132 over 132 into 100. That is basically how percentage is calculated. We have to predict the effect on the heart rate of an increase in temperature to 40. And as you would observe, there seems to have been an increase now. Initially there was a increase, and now there is a increase. There was an increase and now there is a decrease. And if we observe the pattern, initially we may actually calculate the average for these. So the percentage increase has been decreasing. So now it would be a good idea to think, or a good thing to rely on, that the percentage decrease would also follow a similar pattern. But it might actually be a greater decrease, because at these high temperatures it is a possibility that the organisms, enzymes, and the organism is actually dying. So it could be a prediction of it. Or there may actually be a possibility that a similar trend is followed as this, and from 35 to 50 there is actually a decrease, but by a smaller amount, say minus 45. So that doesn't seem all that feasible when you think about the reality, because if this is decreasing at this point, it means that those conditions are not ideal for it. But I would still go with the pattern one. Assess how far the results of the investigation support the hypothesis, and that is that for every 10 degrees Celsius increase in temperature, heart rate doubles. So let's compare. 5 to 10 is 5 degrees Celsius, and 10 to 15. So there was a 50% increase and there was a 40% increase, so almost double. So 52% and 38, again almost double. So to an extent it is right, because there is definite and almost double. A double would be a 100% increase, but here it is a 90% increase, and here it is again a 90%. When we are going from 10 to 20, it is a 92% increase, and from here to here it is a 52 plus 8, 60, 90%. However, beyond this there isn't as much an increase anymore. So to an extent it is right, but afterwards there is actually a fall, because this is suggesting that it will continue to increase, but the fact is that actually it would go like this, then begin to decrease like this, and then it would follow a negative trend, a negative gradient. So let's see. Length of the organism, time for adjustment, time of measurement, activity, age, sex, a mass of organism. Yep. Counting high rates is error prone. Temperature changes or activity. Yep, this was wrong though. Idea of sufficient measurements for reliability. It might actually be better to see what question we are answering. So, two variables that have been controlled, we set both of them. Length of the organism, the time of measurement, and the time of adjustment. And one other variable that could be controlled, definitely mass. Age is a possibility, activity, initial activity is a possibility, sex is a possibility, water, pH, volume, source, oxygen supply, all of this is a possibility. One feature which may cause the results to be inaccurate, because the heart rate is probably pretty high, so we may have some amount of error in this one. And then there is suggest a reason why the student used 5 specimens at each temperature. Idea of sufficient measurements for reliability to remove anomalous results. Why the student converted the raw data to percentage change. Allows for different starting points, can see the changes more clearly. Yep, comparisons, easy to make. Minus 25, graded 25, definitely. And then predict the effect on the heart rate. Decreases by at least 50%, falls to a very low value, or may stop altogether. Possibility. And from here to here, as rate approximately doubles with 10% increase. However, it increases but does not exactly double, so to speak. And after this, there is an actual decrease, and there is a rapid decrease. So this is definitely right. So we get 2 marks for this, 1 mark for this. Let's go with 0.5 for this, and let's go with 0.5 marks for this. 1, 1, 0, and 2. From 10, how much do you get? 2, 3, 4, 5, 6, 7, 8. So in total, from 30, you would have gotten 26 for this if you'd done this, and these are good marks. But as always, we strive for the best, and improvement is something you should always be ready for. Let's open the examiner report. Some students miss reading these, thinking that it's not all that important, but in my experience, they do matter a lot. See, 20, so this was a bit of a difficult paper. Hence why I choose it. Here we are to the examiner report. The candidates' answers showed a wide range of understanding, and there was no indication that candidates were short of time. There were some excellent answers showing the ability to apply knowledge and understanding to unfamiliar context. And those candidates had clearly read the information carefully and made good use of it in their answer. Again, something I emphasized already. Other candidates seem to have misread or misunderstood the intention of the question and so gave poorer answers, and this was particularly evident in question 1C2 and 2A and C. Let's see what they're talking about. Question 1. Yep. And question 2, A and C. Yeah, seemed easy in my opinion. It is important that candidates remember that the paper is based on practical knowledge and experience. Yep. So you need to know how measurements are made and how results are processed. Question 1. The question was intended to assess the ability of candidates to identify the independent variable in an unfamiliar context and describe a variety of measuring procedures using a large sample collected from the environment. Question tested the understanding of statistical testing and drawing conclusions, and candidates showed uncertainty in identifying the independent variable and were often imprecise in describing the methods for measuring. Great deal of confusion between measuring using a microscope reticule and collecting a magnification. And for the first part, candidates tended to repeat the information in the question and refer to sun or shade leaves. Alternatively, one of the dependent variables was sometimes stated, and the expected answer was for some idea of the difference in light exposure of the two types of leaf, either in terms of intensity of light or duration of light. And for the second question, there were some good answers, in some cases exceeding the maximum marking point. Most answers followed the data sequence in table 1.1. Better answers suggested an appropriate method of measurement of all dependent variables plus a sample size of 30 or more to obtain an average or a mean. Poorer answers said to measure without stating a method. Always state a method. A variety of methods were acceptable. A ruler, calipers, see I told you, a string or cotton, all acceptable for measurement of the internodal length. When measuring surface area, candidates need to remember to double their estimated size to take both sides of the leaf into account. Some realized that as they were almost circular, a mathematical formula could be applicable. Poorer answers appear to be confused about the mathematical formula, as they gave the formula for the area of a rectangle or the formula to find the circumference of a circle. Most candidates referred to the use of a balance, and better answers showed an understanding that the individual leaves would have a low mass and weighed the total sample together and then divided to find the mean. Yep, this is something I missed. Most candidates also used a pottometer to measure transpiration. Credit was awarded for description of how the pottometer could be used. A variety of answers were acceptable. For example, measuring mass loss for 1 hour or measuring distance moved in a capillary for a known time period and estimating the rate for an hour. The poorer answers confused a pottometer with a respirometer. And there was also marks available for processing the data. Better answers described how the mean leaf surface area to leaf mass ratio is calculated. Poorer answers tended to say only that the ratio is calculated. Candidates needed to explain how ratios and mean values are obtained. And answers that show formula for calculating a mean were acceptable. And those who recognized that either standard error or standard deviation had been calculated also received credit. And some candidates appear to misinterpret the question or this try to group plants. Answers were variable for B1. Better answers gained full credit by filling in the correct figures from the information given. This was easy. 2 was also variable. Better answers showed the understanding that there were 2 sets of data, each with 30 values which needed to be taken into account. Poor answers did not gain this and often gave the answer 59 minus 1 or n minus 1. Many candidates gained full credit. Better answers refer to both t values being greater than the critical value. Yep, this was in terms of finding the magnification rather than the actual thickness, which was quite expected. And here they were relatively good answers, but answers related to the adaptations to limiting water loss or to maximizing light absorption. And this is it for this video. Hopefully you found it to be helpful.